Harshal, I'm a junior here at McNair. So today I'll be sharing with you a little bit about my research. So harmful algae blooms.
So hold it really close to me. Yeah,
Okay, sorry.
Talk right into it, yeah, I know.
Okay, so algae are these microscopic organisms that can be found in nearly any aquatic body. So algae can range from anything to being small single-celled organisms to large thick mats. So harmful algae blooms are basically just when you have a lot of algae. And so as you can see by this map, most of the US has a massive problem with harmful algae blooms, and it's not just the US, even worldwide, algae blooms are just becoming a more and more prominent problem. So algae blooms are harmful because of three reasons. So a lot of species of algae, they release toxins into the body of water that they inhabit. That, along with hypoxia, just devastates a body of water when there's a harmful algae bloom. Furthermore, they're also a very expensive problem to clean up because these toxins are so small, to filter them requires millions and millions of dollars. So harmful algae blooms largely occur because of two things, nitrate and phosphate. So when farmers apply fertilizer on their field, what they're basically doing is they're applying nitrate and phosphate. When it rains, all that excess nitrate and phosphate ends up going downhill into a body of water, and that nitrate and phosphate, which is meant for the plant, ends up feeding the algae, causing for the algae to grow, and thus we have a harmful algae bloom. So the long-term goal of my 5-year research is to develop a cost-efficient, effective, and most important of all, eco-friendly way to prevent harmful algae blooms. So I started this back when I was in 8th grade, which was the first phase of my research. So in the first phase of my research, I discovered that Stropharia mycelium, which is the root structure of the Stropharia mushroom, could remove nitrate and phosphate from water. So this was a very elementary experiment, and this is just my, this is my room, and I set up these trays in my room, and I was able to test my samples using very easy to buy test kits, but my results were not the most scientifically accurate. So next year, when I came into McNair, I spoke to Mr. Stanton, and this actually is a picture of his room. It's one of the tables up near his desk. So in 9th grade, I was able to set up about 60 or so of these funnel systems, and using the equipment we have at McNair, such as spectrophotometer, nitrate ion selective electrode, I was able to get much more accurate results. So after phase 2, which was 9th grade, I went on to phase 3, which is 10th grade. And in phase 3, I further expanded upon my project by using more complicated systems of filtration, and I was able to get funding from the school, especially Ms. Tribona, who bought me a nitrate ion selective electrode, which just gave me just amazing results. And because of the third phase of my project, I was able to compete at the International Science and Engineering Fair, so that was also an amazing experience. So this is what my project looked like. This is the conference room next to Ms. Osaria's room. So the school was kind enough to let me make a mess in there and do my project. And this is what the other system looked like. So currently I'm in the 4th phase or 4th year of my research. So because of my success at the science fair last year, a professor at Montclair State University, Dr. Yang Deng, he reached out to me and he's letting me use his labs to conduct my experiment and test my samples. So upstairs right now on the 4th floor, I have about 40 or so of these trays set up, and every weekend I go to his lab to test my samples and get nitrate and phosphate readings. So yeah, thank you for your time, and if you have any more questions, you can visit my website, havprevention.weebly.com, and I'll also be in the back there with my board if you have any more questions. Thank you.